Hi, my name is Dr. Shatrughan and we are going to discuss a topic cellular aging. It's a very small topic, so it will take only 15 minutes. Please watch the video carefully. What is cellular aging? The cellular aging, it is a progressive decline. It is a progressive decline in cellular function and its viability. Both cellular function and viability decreases rapidly, and the basic cause behind all these things is genetic. There is some kind of genetic abnormality which causes cellular function and viability uh, decrease. Okay. The next is there is accumulation of cellular molecular damage. There is a lot of damage. If there is a lot of damage, the cell will die or cell will age and then die. These are due to exogenous interference. There some exogenous products may damage, and when they accumulate in cell, just like ROS, reactive oxygen species, or any kind of free radical. So we got to know two reasons of cellular aging. First is genetic abnormality. The example is any kind of mutation. The other thing, exogenous influences, or I can say free radical injury. These both can cause cellular injury, and this is the basic definition of a cellular aging. What are the factors affecting cell aging? The factors affecting cell aging, so the first is DNA damage. As we have discussed in the definition, there is some genetic component or genetic abnormality, and it will lead to DNA damage. The damage of DNA can be due to exogenous or endogenous causes. Mostly exogenous causes we consider carcinogen exposure, like arsenic, heavy metal. They are very prone to cause cancer, and they causes cell damage and DNA damage at nuclear level, which will lead to cellular aging. Endogenous are, I have already told you, reactive oxygen species. Okay, free radicals, carcinogens, and ROS. There are three syndromes. Okay, all the three are associated with the increased rate of aging. Okay, these three syndromes, Werner syndrome, Bloom syndrome, and ataxia-telangiectasia, all three are associated with an enzyme defect called helicase or other replication enzyme defect, because the DNA cannot is not able to replicate. So uh, either it is not able to replicate, or the repair of that replicated DNA or the DNA is not possible. These both are the reason that DNA damage will lead to senescence or cellular aging. So Werner syndrome is what? It is a premature aging, which is due to a defect in enzyme called helicase. Helicase is an enzyme of DNA replication. Okay, or it may hasten the DNA repair process. The other syndrome that which I am suggesting is Bloom syndrome. Bloom syndrome is an autosomal recessive disorder. Similarly, premature aging is there in that, associated with short stature in presentation. You have to remember this. Okay, there are many features of Bloom syndrome, but the important one I have mentioned here is short stature. You need to remember it. The next is ataxia telangiectasia. Ataxia means loss of balance. Okay, you will get the hint here. Ataxia telangiectasia is also due to DNA replication enzyme or DNA repair enzyme defect. All three causes are associated with early cell death or early cell aging. The next cause is cellular senescence. Senescence mean senescence. What does senescence means? Okay, senescence means the cell is reaching its terminal and non-dividing state. I am saying non-dividing state. How do we define that cell is at its non-dividing state? Okay, so it is done by two procedures. First is telomere attrition. The next is tumor suppressor gene activation. I will make you understand both of these. So the first is telomere attrition, that is progressive shortening of telomeres. So first we should know what are telomeres. If this is a chromosome, at the last joining point, the base pairs at the last point, these are a band which we called telomere. They are useless bands, okay? They don't code for anything, they are just there for it. There is a concept you need to understand that in every and each and every replication, there is some loss of DNA, and that loss of DNA is from this part, terminal part, okay? Then the terminal part is lost. I am going to suggest you, if there comes a time when this terminal part is not there, at that moment the cell lose their ability to replicate. And that is what senescence means. That is what telomere attrition means. That is progressive shortening of telomeres will lead to the cell lose his ability to replicate, which is cell cycle arrest stage is there. So what is the limit? Okay, there must be a finite limit, na, that okay, the cell will divide. How long it will divide? So it is defined by a limit. We call it as Hayflick limit, which is actually 40 to 60 times. Hayflick limit is a identity defined for a somatic cell, which is a normal cell. There is an enzyme, okay, telomerase. It is an enzyme which maintains the telomere length. If this is cut in every replication, this enzyme, what it will do is it will make newer strand. Okay, the length will be maintained here. So the activity of telomerase will define whether the cell will die or reach its terminal non-dividing state or not. Okay. So what type of cell and telomerase activity? I have made a table. Okay, let's understand what I want you to know about this. Say there is a germ cell. Okay, the germ cell has work to divide. Okay, say this is a germ cell. It will divide into two daughter cells. These daughter cells will divide again. Both daughter cells can divide. Okay, they both have potential. So this will go on. We know this. Okay. So here the telomerase activity is very well expressed. Okay. It has a continuous, it need to continually divide, okay, with the same potential. So we need telomerase activity. The next is stem cells. There is a unique thing about stem cell. Let's say this is a stem cell. What it will do is, this is one is a stem cell. It will divide. First is one daughter cell. Other is again one stem cell. This daughter cell will do its function and it will go on to division further. But this stem cell will further divide into one daughter cell and one stem cell. So there is a single stem cell here and the rest other part is normal multiplication just like germ cell. So it is expressed but it is lower in quantity. The telomerase activity is lower in stem cells. Let's say there is a somatic cell. The concept here is there is a somatic cell and somatic cell do not divide. So there is no telomerase activity needed because it why it does not divide, because it has got no telomerase activity. It can divide up to a certain limit and after that it will not be able to divide. That certain limit is 40 to 60 times, which is called Hayflick limit. The next is a cancer cell. The cancer cell has done what a cancer cell is. A somatic cell or any kind of cell, germ cell, stem cell, but I am taking an example of somatic cell. And a somatic cell keeps on getting proliferated. That means he can divide unlimited times. For division, he needs telomerase activity, but there is no telomerase activity as per se in normal somatic cells. So, what will be the status of telomerase activity in cancer cells? We'll say reactivated. Why I'm not saying expressed or anything else? Because there was no there was no telomerase activity in before. It is a reactivation of a activity. Okay, telomerase enzyme activity is reactivated in them. Two diseases you need to know. These are given in your textbooks of pathology. Pulmonary fibrosis and aplastic anemia. They are associated with shortening of telomere lengths. See, shortening of telomere lengths are associated with chronic and very dangerous disease. One is pulmonary fibrosis. The other is aplastic anemia. I hope you have understood the concept of cellular senescence. This is the second pillar of cell aging. The first is DNA damage, the other is cellular senescence, the natural death of a cell. Next, I will discuss is defective protein. We are coming from center to outside of cell. Okay. The third factor which defines the on which cellular aging depends. The third is protein homeostasis. See, homeostasis means balance. So there are two identities we need to know. First is chaperones. I will discuss a little bit about chaperones. Chaperones are heat shock protein family. Okay, heat shock protein family. There are various numbers. Their main function is to maintain the level, correct folding of proteins. If there is a protein, you have heard of structure of protein. They can be primary protein, secondary protein, tertiary protein, quaternary protein. Do you ever, you should wonder how these folding occurs. The main family of protein, heat shock protein family, is the main identity responsible for the folding of these protein and making them function into larger and complex structures. The thing you need to remember is chaperones are the family which folds the protein. Let's say if there is an error in folding that protein, so it will not function regularly, and we need to degrade that protein because it is not of our use. So two degrading system, I want you to listen to this. uh, First is autophagosome and lysosome system, the other is ubiquitin proteasome system. Autophagosome, lysosome system, and ubiquitin proteasome system, their function is to degrade. Yes, their function is to degrade the misfolded proteins. So these both chaperones and ubiquitin proteasome system are the main significant part of protein homeostasis. And if that protein homeostasis is very well defective, see both these are a main contestant of impair. If both are impaired, the maintenance or degrading system, they will cause cellular aging. Okay. The last one is deregulated nutrient sensing. I will just you need to remember one line I am gonna say here. The only proven method to delay cellular aging is calorie restriction. If you eat less, you will live long. That is the dictum here. If you eat less, you will live long. The only method to delay cellular aging is to eat less. Let us discuss why I am saying that. There are two things we are going to discuss. The first is insulin and IGF-1 signaling pathway. What is it? I'll tell you in briefly. If I eat glucose, let's say I eat food, we will understand why I am saying calorie restriction. Calorie restriction is the only proven method to delay aging. We eat food and the glucose will enter, and glucose will stimulate a hormone called as growth hormone. Growth hormone in turn stimulate liver to produce IGF-1. IGF-1 is what? IGF-1 is insulin-like growth factor 1, and it will do some anabolic shit. Anabolic function are performed by two particular function in Robbins. You can see what is written, a kinase enzyme AKT and mTOR. mTOR is an enzyme stimulated which is of mammalian target of rapamycin. It inhibits the aging. Okay, it's rapamycin inhibits cell aging. So mammalian target of rapamycin. If we eat more, this will function more and the cell will die early. If we eat less, the lesser amount of IGF will be secreted, the lesser anabolic function will be there, and a lesser cell aging will occur. The other part we are going to discuss, which is very important, these things are called sirtuins. Sirtuin is an NAD dependent protein. Each and every line is very important. It's a short note. NAD dependent protein, not an NADP dependent protein, NADH dependent protein. It's a NAD dependent protein. It's not a gene. The gene for sirtuin is SIRT gene. See, sirtuin is a protein and SIRT is a gene. Uh, what is the enzyme? Okay, this protein. What is the enzymatic activity it does? What is its function? It is histone deacetylation. That is histone deacetylase. That means it works on deacetylase histone. Histone is a part of DNA. Okay, so it works on chromatin level. What it does is ultimately it decreases the glucose requirement. What, in other words, glucose requirement will be decreased. That is equivalent to what? Calorie restriction. So sirtuins are a potential candidate for decreasing cellular aging. It decreases cellular requirement of cell, but how does it do it? It increases the insulin sensitivity. It is a very important thing to know. Sirtuins increases insulin sensitivity. And if the cell will not age, then cell will not die, and cell death, which is programmed, is called apoptosis. So the apoptosis will not happen, and that will be decreased. Cell repair is opposite to apoptosis, and it will be increased. Other thing is it will stimulate telomerase activity. All the things good we need for the cell to live long. Okay. There is one thing you must know. The wine, particular alcohol, are found to have a certain kind of certain in them. So this is the topic of cell aging. Four main factors. We have discussed all of them. Kindly read your standard text of pathology for this topic. Thank you for watching.